Hello everyone, welcome to my presentation on Archimedean NanoMIP sensor, a prognostic device for clotting disease detection. Hemostasis is a natural process to control the bleeding of an injury by activating the clotting cascade. Any protein deficiencies in this hemostatic system causes hypercoagulation, which produces excessive blood clots in human body. Abnormal blood clots in human body leads to chronic diseases such as heart failure, stroke, deep vein thrombosis, and pulmonary embolism. According to WHO, these non-communicable diseases are the prime cause of mortality. Early detection and timely treatment are essential to meet the global target set by the United Nations Sustainable Development Goal to reduce the mortality rate from non-communicable diseases. In compliance with this, we will be able to provide a digital healthcare monitoring system with enhanced biosensing on clotting syndrome. This research work has international collaborators, a medical venture innovation company from Penang, Malaysia, and the First Center Hospital of Baoding City. The patent for this innovation has been filed under the Intellectual Property Corporation of Malaysia. We have successfully published more than 10 ISI journals. Here are the recent publications. We have secured several research awards with distinction. During any surgical procedure, maintaining the delicate balance between bleeding and clotting is critical. A patient with hypercoagulation factor holds a higher chance of life-threatening complications when they go through the surgical treatment. Clotting assay is a pre-operative procedure for a surgical patient to address the blood clotting level. This assay measures their blood's ability on how long it takes to clot. Based on this assay, anticoagulant medications will be injected on the patient before surgery to keep the patient's blood uncoagulated along the operation. Existing clotting assays are laborious, time-consuming, costly, and requires large sample volume. So what we need is a rapid and sensitive point-of-care monitoring system for clotting diathesis. We designed a prototype for digital blood clotting monitoring system for 3 different types of sensor pattern to facilitate amperometric, voltammetric, and imperimetric detection strategy. Following simulation video presents an overview of the device functionality and the components. The protein level sufficiency will be indicated by the green light for sufficient and red light for insufficient. The concept of the invention considered 2 main ideas. The first idea is the transducer material and design, where we used interdigitated Archimedean spiral pattern. The second idea is enhancing the sensing material by artificial receptors, which is molecularly imprinted polymer. Conventional photolithography technique was used to fabricate the Archimedean sensor. The novelty of the developed biosensor is conjugation of carbon nano-horn and gold nano-urchin as hybrid materials. Here we summarize our invention. This product is suitable for flexible detection and quantification of human protein level without pre-treatment step. Due to the nano-gap design, it requires only a small amount of test fluid like single droplet. The chemically and physically robust imprinted polymer conjugation enables repeatable use of the sensor. This device is compact and handy for point-of-care detection with multiple detection strategy. Alternatively, the probe station and the processor components can be directly connected to the lab instrumentations for high-throughput analysis by just plug-and-play connection. Imprinting technology grants rapid detection in less than 5 minutes. The marketing targets for this innovation are coagulopathy detection, pre-surgery procedure, dental extraction care, and hemophilia B detection. This invention is non-hazardous because it is reusable and it requires only droplets of test fluid. Moreover, no sample pre-treatment is needed, so bio-waste is completely minimized. Existing protein assays such as BCA assay, Lowry protein assay, enzyme-linked immunosorbent assay are only able to detect from millimolar to picomolar level. Our sensor is able to detect up to femtomolar level at lower price compared to them. Thus, our research has proven simple, inexpensive, non-hazardous, and portable diagnostic setup with higher sensitivity and rapid detection at bedside analysis, also for early-stage clotting disease diagnosis. An early diagnosis is a better disease management that can be an excellent economic investment because if provided early to patients, they can reduce the need for more expensive treatments.